Hello everyone. Welcome to the new session of Animal Kingdom. So today we are going to study about the first session for this particular chapter, right? So this is the introduction part. So when you look around, you will observe different kinds of animals with a different structure and form. So our earth is full of so many animals. So we are having around 1.7 to 1.8 million of a species, right? And it is very difficult to study each and every species. So if you want to study about so many species, then it will be very difficult, right? So there is a classification system, right? There is a classification system in which the and different same type of animals which is having the similar characteristics has been grouped into a one particular phylum, right? So this is the way to study all these animals. And when you observe so many different animals, their structures and your forms, right? So you must be observing that how to describe all those animals, how to study about all those animals, right? So as over a billion of species of animals have been described till now, the need for the classification become all the more important. So it is very like important to classify all those organisms so that our study become easy. We can describe each and every animals. We have a data for each and every animals, but not individually on the basis of their groups. Okay, so just by seeing the animal, you can say that this particular animal belongs to this particular phylum because you know the general characteristics of those particular animals as well as the phylum, right? So it is this is the way to describe all those animals. So as over a millions of species of animals have been described till now, the need for classification becomes all the more important. So that is why to study all those organism on the basis of classification is a very important nowadays. The classification also help in assigning a systematic positions to the new described species. So you can see this we are having just starting from the vertebrates. If we talk about the vertebrates, then these are the phylum we are having now. Porifera, then Nidaria, Nidaria, then then after this, we are having the phylum of the worms. Okay. Then comes the platyhelminthes. No, there is one more phylum. Uh, that is Ctenophora. Ctenophora. Then comes then we are having platyhelminthes, helminthes, then esculenthes. Okay, esculenthes. Then we are having a phylum of the earthworm, right? Annelida. Annelida. Then we are having arthropoda, right? Then we are having echinodermata, echinodermata, right? So these are the name of the phylum, right? And if we know like where you will keep the planaria, where in the which phylum you keep in the platyhelminthes, right? Because it is a flat worm. Okay, flat worm. Platyhelminthes means flat worms. So now you know the characteristic of this particular phylum, right? So you can keep that particular organism accordingly. Okay. So the classification also help in assigning the systematic position. So in this way, we can assign the systematic position. Suppose you find an X organism. Okay. Now this is a segmented one. Segmented. When you see that particular organism, you see that it is having segmented and it is worm. So you keep in the segmented worm, right? They are having closed type of circular system. When you open that organism, right, you see that they are having closed closed circulatory system. You see that they breathe with skin, breathe with skin. Skin is moist. So where you keep all these this organism X1, you keep it in Annelida, because all these characteristics of this organism X is matching with the phylum Annelida, right? So in this way, you can assign some of the characteristic. When you observe that organism, you write some of the characteristic, and now that characteristic can be matched with the existing phylum, and you can keep that particular organism in that phylum. So this classification also help you assigning the systematic position to a new described species. So this is suppose the new species, you can keep it in the Annelida because it is having certain characteristic and that characteristics has been matched with the phylum Annelida. Now, in spite of differences in the structure and form, different animals, they are fundamental features common to the various individual in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive and circulatory or reproductive system. So there are so many organisms. Each organism differ from the first one or the third one with the some fundamental features, right? So what, on the which basis you can differentiate them? You can differentiate them how their cells are arranged. When you see a particular organism, suppose there are three organism X, Y and Z. Okay, now the characteristic what you are following to see them: arrangement of cells, arrangement of cells, whether they are multicellular, they are having the tissue grade of organization, whether they are having the organ grade of organization, right? You see that. Now, body symmetry. What is the body symmetry of that organism? Second characteristic you follow: body symmetry. Body symmetry. Whether they are bilateral, whether they are lateral, right? So you talk about the symmetry, you see the symmetry of that particular organism. Coelom. Next is the coelom. You look about the coelom, that is the body cavity. What type of body cavity they are having? Body cavity. Now, you also see the patterns of digestive system. Patterns of digestive system. You see their circulatory and reproductive system. Circulatory and then reproductive system. So these are some of the characteristics that you see, okay, that you see when you see three, four different organisms, how they are different and in which phylum you can keep them. On the basis of, because every phylum has its own characteristic. Now you find some animals and you see what are the basic characteristics of that animal. And according to that, you can keep that animals in a particular phylum. So these features are used as the basis of animal classification, right? So these are some of the bases that could be used. This is the basis of classifying the animals. So let us see some of these characteristics in details. The very first one is the level of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. Whether you or talk about the porifera, for example, you can talk about Sycon, Euspongilla, right? They are having what arrangement of the cells in their body. Okay, now here you are having you can talk about arthropoda, arthropoda, or you can talk about echinodermata, you can talk about mollusca, right? So all the animals are multicellular, whether there is a prorifera, whether there is arthropoda, whether it is echinodermata or mollusca, all are multicellular and they do not exhibit the same pattern of organization of cell. Their organization of cell is different in each phylum. So, for example, sponge, the cells are arranged as a loose cell aggregates. They are multicellular, but they do not have they do not have a tissue grade of organization. Still, they are multicellular, but no tissue is formed in the case of porifera. No tissue formation. You cannot see any type of tissue, no tissue. So this exhibit the cellular level of organization. This is very, very important. Porifera is multicellular. Still, it is showing the cellular level of organization, cellular grade of organization. Okay. So some division of labor activities occur among the cells. So how they divide their all the activities, how they divide their functions, they divide their functions on the basis of their cellular activities. So you can see here they are having spongocele cavity like this. Suppose this is a sponge, right? And they are having small opening all over their body surface like this. And this opening lead to the inner side. That is the spongocele. This is the spongocele cavity. Spongocele cavity. Now, from this ostia, and these openings are called as ostia. Now, water move from this to inside. Okay, they are having here a special type of cells. They are having all their surfaces surrounded by a collar cells, coinocytes. Very important. Okay. Now these coinocytes take away the food material from this which comes from this water, from this ostia. The water moves inside the spongocele cavity, and this spongocele cavity is having the lots of oxygen dissolved in the water and the food material. So that is taken by these coinocyte cells or the collar cells. So here the division of the labor we see, okay, division of the labor, that is the activities we see among the cells. So in coelentrates, now if you talk about the coelentrates, the arrangement of cell is more complex. As you move from one phylum to another, the complexity is more. They are more advanced than the previous one. So they are having the new types of arrangement of cells, and that is quite complex if you compared with the poriferates. Okay. Level of organization. Here the cells performing their same function and are arranged into the tissue, and tissue hence is called as the tissue level of organization. So at the next level you see the cellular level of organization in the case of porifera. Now here you see the tissue level of organization. It means that different cell aggregates together, they come together and now they are working as a tissue. So you see the tissue level of organization, and that is the advancement. That is the advanced feature or the characteristic of that particular phylum, right? A still higher level of organization, that is organ level, is exhibited by the members of platyhelminthes. When you reaches to the next phylum, platyhelminthes, okay, after the ctenophora, you see that they are having different organ. For excretory system, they are having the flame types of cells, okay. So they are having a different reproductive system, right? So they are having different reproductive organ for that. They are having mouth, they are having the suckers, right? So now they are having a organ level of organization. Now this is again one head advanced feature than the tissue level of organization, which we have seen in the case of Nitaria, right? So this is again the advanced feature. Now, and other higher phyla where the tissue are grouped together to form the organ, each specialized as a particular function. So they are having the organ level of organization. Now, in animals like annelids, arthropods, mollusks, echinodermata, and chordates, organ have associated to form functional system. Now, they are different organs that comes together and perform one particular function because they are forming a particular system. Now, they are having particular system, circulatory system. They are having excretory system, okay. They are having nephridia, okay. Now they are having digestive system. So different for different metabolic activities, they are having the different. For circulation, they are having circulatory system. For excretion, they are having the excretory system. For digestion, they are having the digestive system, right? So in the these are the phylum in which we can see the organ system level of organization. Here you see the specific system for the particular activity. Now, this pattern is called as the organ system level of organization. So, organ system is different groups of animals exhibiting various patterns of the complexities. Okay. Now, this organ system is having different groups of animals and they are showing some level of complexities. Very complex type of things you can see in the case of the organism which is showing the organ system level of organization. For example, the digestive system in platyhelminthes has only a single opening to the outside of the body and that serve as both mouth and anus, and it is called as incomplete. Digestive system is incomplete. We say that digestive system is complete only when it is having two openings, one mouth and another is the anus. Then we can say that okay, this particular organism is having the complete digestive system. But here in the case of platyhelminthes, you see only one opening, okay. You see only one opening and that is the mouth. No anus is you can see in the case of platyhelminthes, right? This is the one of the peculiar feature, and according to that the digestive system is called as incomplete. The complete digestive system has two openings, that is having both mouth and anus. Similarly, the circulatory system be of two type. Now, if you see the different organ system, then in the organ system only you can see there are the different types of organ system. If you talk about the circulatory system, then circulatory system can be of two type: closed, closed and open. Very important, closed and open type of circulatory system. Open, first one is the open in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it. So when the blood is pumped, okay, out of the heart. It take out of the heart. The blood is taken out of the heart and it is given out to the tissues. Means all the cells and tissues are bathing in this particular blood. This is called as open type of circulatory system, as we see in the case of arthropoda. Now coming to the next level, that is the closed type, okay, in which the blood is circulated through a series of vessels of varying diameter. Okay, we are having the different cap this vessels. Now these vessels can be arteries, veins and capillaries, as you see in the case of annelida. They are having the closed type of circulatory system, right? But in the case of arthropoda, we are having the open type of circulatory system, right? In the case of human also, we are having the closed type of circulatory system, because we are having arteries, the one that carries the oxygenated blood from heart to the different organ. Okay, veins, the one that carry the vessels that carry all the deoxygenated blood from the different body parts, and it takes toward the heart. Heart gives it to the lungs, where it get purified and it comes back to the heart. Now there is a third type of vessels that is called as capillaries. Its lumen is very thin, and it is less than the arteries and veins both. So when arteries reaches to a particular organ, it divides and redivides into fine capillaries. So these are the capillaries. In the same way, veins, okay, different veins coming out from the different organ or particular one organ, but the different parts of that organ, they comes and unite together and form a one particular vein. So suppose this is an organ, okay, here the veins, the capillaries are coming out from the different parts of the organ and uniting together to form a one particular vein. So suppose this is a liver, then this will becomes the hepatic vein, right? It is taking all the deoxygenated blood from the organ. So that is the vein. And what happens, suppose this is a tissue, suppose this is any tissue or an organ, then what happened? When the artery reaches to that organ, it divides into fine blood capillaries. Fine blood capillaries, like this. So here it is giving out in the different branches, that is forming the capillaries. Okay, this is the capillaries, and here capillaries unite together to form a vein. So this we can see in the case of closed type of circulatory system. I just want to tell you that the blood is not released like this only when it reaches to the tissue or organ. It enter in the tissue and organ via these capillaries and taken out from this that particular organ with these capillaries only, right? So this is the blood is not given out like this only. It is not given, as you can see in the case of open, when the blood reaches to the organ or to the tissue, it is released that way and the tissue and the cell can bathe in it. So this is not the way in the case of closed type of circulatory system. Next is the symmetry. So animal can be categorized on the basis of their symmetry. We can categorize animal on the basis of their symmetry. Okay, so let us see about what is symmetry. Sponges are mostly asymmetrical, and in any plane that passes through the center does not divide them into equal halves. So there are three different types of symmetry we find. Asymmetrical. Asymmetrical. Then we are having bilateral symmetry. Bilateral symmetry. That we are having radial symmetry. Now, suppose any organism is like this. Okay. If you divide this organism from this this part of the organism is not giving the same mirror image of this. Okay. So when you are not able to divide organism into two equal halves, okay, two equal halves, then it is called as asymmetrical. So the poriferans, okay, the sponges are mostly asymmetrical. That you divide it any plane through the center, and it is very important that it should pass through the center of the body. It does not divide them into equal halves. And when any plane passing through the central axis of the body divide the organism into two identical halves, it is called as radial symmetry. So suppose any organism like a starfish is there, or any organism like you can see the ctenophora also. Okay, ctenophora, you can divide the organism from the center like this. You can divide from this plane also. You can divide from this plane also. You can divide from this plane also. So number of plane you can divide the organisms into the two equal halves. Okay, two identical or two equal halves. This is called as radial symmetry. This is called as radial symmetry. Now, the coelentrates, ctenophores, and echinoderms have this kind of body plan. So here you can see the radial symmetry. This is, we can say there's any need area. Now, we pass one of this uh, cutting. If we cut this organism in this way, then we can get the two equal halves. Okay. Similarly, if you cut from this way, then again you can get the two equal halves. So there is a number of plane through which you can divide the organism and you can get the two equal halves of that particular organism. So that is showing the radial type of symmetry. That is showing the radial type of symmetry. Animals like annelids, arthropods, where the body can be divided into identical left and right halves in only one plane and exhibit the bilateral symmetry. So this particular this is a plane through which you can divide this crab into two equal halves. Okay. So here you can see this is having the one pair of leg, then simultaneously the paired one is here. For this, it is here. For this, it is here. For this one, it is here. For this clause, this is the clause, and this body part is totally equal to this particular part, right? So we can say that this is the two equal halves. Now, if you can divide the organism if like this way, then are this and this are the two equal halves? No. So there is just one plane that you can divide the organism into two equal halves, right? So that is called as bilateral symmetry. So what are the phylum? Annelids, arthropoda, etc. These shows the bilateral type of symmetry. Okay, now let us see the diploblastic or the triploblastic organization. So this is the, you are having the arrangement of the cell at the time of embryo formation, and these cells, embryonic layers, okay, forms the different organs in the body, and that is fixed. Like ectoderm will form this particular organ, or mesoderm will form this particular organ, or the endoderm will form this particular organ. In this way, the organs that has to be formed from the particular type of embryonic layer, that is fixed. So animal in which the cells are arranged in the two embryonic layer, only two embryonic layers are there, that is the external ectoderm and internal endoderm. it is called as diploblastic animal. Only two layers. No third layer is present, right? So that is called as coelenterate. We can see this diploblastic type of arrangement in the case of coelentrates. Okay, so diploblastic and triploblastic, an undifferentiated layer, that is mesoglea, is present between the ectoderm and endoderm. So here you can see this is the ectoderm, the outer layer of the cell arranged in one fashion, and that is called as ectoderm. Now you can see this endoderm layer, this particular layer. This layer is called as endoderm, inner layer, right? In between the ectoderm and endoderm, there is a layer. This is not a cellular layer. This is a type of, you can see, a substance that is present in between the ectoderm and endoderm, and that is called as mesoglia. That is called as mesoglia. Instead of mesoderm, they are having mesoglia. So this is showing the diploblastic level of arrangement. So those animals in which the developing embryo has a third germinal layer, that is mesoderm, in between the ectoderm and endoderm, are called as triploblastic. Okay, now the organism is called as triploblastic because they are having the third layer now. Ectoderm outerward, then comes the middle layer, that is the mesoderm. Now here it is a special cell layer. It is not just a matrix substance as we have seen in the case of diploblastic having the mesoglia. Okay, it is not like that. So they are having the third germinal layer, that is called as mesoderm, present in between the ectoderm and endoderm, and that is called as triploblastic. So that we see in the case of platyhelminthes to echinochordata. Okay, from platyhelminthes to echinodermata, we see the triploblastic layer. Now comes the coelom, that is the body cavity. What type of body cavity the organism is having? What type of body cavity the organism is having? The presence or absence of cavity between the body wall and the gut wall is very, very important in the classification. So suppose this is the body wall and here it is the digestive tract. This is the digestive system. Digestive system is having the particular tract. This is, suppose this is esophagus, then here this is the stomach, then comes the intestine, like this. Okay, so this is the digestive tract. Now this is the body wall, and this digestive tract is also known as gut wall. Now in between this gut wall and body wall, there is a cavity. Cavity means empty space. This is called empty space or the cavity. Now, the body cavity which is lined by the mesoderm is called as coelom. Now, this is the cavity which is called as coelom. Coelom. This body cavity is called as coelom. And if it is lined by the mesoderm layer, then this is called as true coelom. So the animal possessing coelom are called as coelomates. So those organisms which are having this body cavity, they are called as coelomates. For example, annelids, mollusks, arthropods, echinoderms, hemichordates, and chordates. All these organisms are having they are possessing the body cavity. They are possessing the body cavity, and that is why they are called having the true coelom in that. So some of the phylum that showing this type of true coelom, that is called as chordates, annelida, mollusca, arthropoda, echinodermata, hemichordata, and chordates. Okay. Now, in some animals, the body cavity is not lined by the mesoderm. Very, very important. When it is lined by the mesoderm, all the organisms are called as coelomate organism. That is the true coelom. Okay. Now, instead of the mesoderm, it is present as the scattered pouches in between the ectoderm and endoderm. So the mesoderm is layer, but it is not in the form of cavity. It is the cavity is present in the small pouches, and it is not lined by the mesoderm. So here you can see such body cavity is called as pseudocoelom. Very, very important. Such body cavity is called as pseudocoelom. So here you can see the one pouch, second pouch, third pouch, fourth pouch. Okay. And this is the cavity here. So this cavity is called as pseudocoelom. So the animal possessing them are called as pseudocoelomates. For example, Escalmanthes. Very, very important. Escalmanthes, roundworms. For example, Ascarius. That particular phylum is having the pseudocoelomate. You have to remember this. Pseudocoelom means false cavity. Okay, that is found in the case of Escalmanthes. The animals which the body cavity is absent, no body cavity is there, that is called as acoelomate. For example, Platyhelminthes. Platyhelminthes is not having any type of body cavity. Means the body wall and the gut wall is not having any cavity in between. So that is why it is called as acoelomate. Now comes the segmentation. Segmentation. Now, in some animals, the body is externally and internally divided into segments and with a serial repetition of at least some organ. Okay. Now, here what you can see the segment in the body like this. Now, you see that in this particular segment, some organs are repeated. Okay, organs are repeatedly present in these segments. So in some animals, the body is externally and internally divided. You can see that there is a demarcation on the surface of the body, internally as well as externally. Okay, so this is a serial repetition of at least some organ, and inside when you see that every segment is having the repetition of one particular organ. For example, in earthworm, the body shows this pattern called metameric segmentation. So this is the true type of segmentation when you see the segmentation from outside as well as inside, then it is called as metameric segmentation. And this particular phenomena is known as metamerism. Very unique characteristic of this phylum, that is the Annelida. Okay, so Annelida example is one of the example is earthworm, another is the leeches. And when you see these organism now, you see segmentation. There is a demarcation at the surface of their body. It This demarcation is present from outside. You can see from the outside as well as inside. Now, there is a repetition of one or two organs in this segment. Now, in the case of earthworm, you see the excretory organ, that is nephridia, in these segments. As it is written here, there is a serial repetition of at least one or some organs. So this earthworm, they shows the body pattern that is called as metameric segmentation. So such type of the segmentation that is from outside as well as inside, then it is called as metameric segmentation. Sometimes there is a segmentation that is from just outside but not internally, right? So when it is present from inside and outside both, then it is called as metameric segmentation. Presence of notochord. Next particular characteristic is the notochord. So the notochord is a mesodermally divided rod-like structure formed on the dorsal side during the embryonic development in some animals. So if suppose this is an animal, then you can see this dorsal notochord like this. Dorsal notochord. Now animals with the notochord are called chordates. All the organism in which you find the notochord, it is called as chordates, and those animals which do not form these structures are called non-chordates. Okay, from porifera to echinoderms we are having the non-chordate. It means that notochord is not present from the porifera to the echinoderms. The broad classification of animalia is based on the common fundamental features as mentioned in the preceding section is given. So here we can see, right? This is the kingdom. This is the level of organization. This is the symmetry. This is the body cavity, and this is the phylum. So let us see here what we see here. All animals are multicellular. Yes, all animals from porifera to chordata, all the animals that belongs to this animal kingdom are multicellular. Now we see the two levels of organization: cellular level and tissue organ or organ system level of organization. So the one that is having the cellular level, it is totally different, and it is only one phylum that shows the cellular level of organization, that is the porifera, right? Now coming to the symmetry, if you see the symmetry in the case of porifera, no symmetry is seen, no symmetry is seen, and they are having acylobate, means no true body cavity is found. They are having the cavity, but that is not the true cavity. Acoelomates. Okay, so these are not showing any specific characteristic. Like here you can see the cellular level of organization and level of organization, and they are having the mostly asymmetrical, that is symmetry is asymmetrical, then they are having no body cavity. So this is a very, you can say, a lower grade of characteristics, and that is why this porifera is the first phylum, the the phylum that is not having even the basic characteristics, right? Now let's see the organism that is having the tissue or the organ system level of organization. So from the cylindrata and from the chordata, you can see all are showing the tissue organ or the organ system level of organization. Now, if you talk about the symmetry, then the symmetry that could they are having, that is the radial and bilateral. Now, in the radial, okay, they are showing the two different phylum, that is the Cylindrata and Ctenophora. Now, the one that is showing the radial, it is also showing the acoelomate body cavity. Still, at this body cavity, they are not so advanced. Okay, Cylindrata and Ctenophora both do not have the body cavity, but they are showing the radial level of symmetry. Now, the one that is showing the bilateral symmetry, we can see from the platyhelminthes and to the chordata, the organism can be divided into two equal halves from one central line. So one that is showing without a body cavity acoelomate, that is the platyhelminthes, with false coelomates, that is just one phylum that is showing the pseudocoelom, that is the false coelom, that is Escalmenthes, and after that we are having all the organism with showing the true body cavity, that is from the Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata, right? Now the Echinodermata exhibit the radial or the bilateral symmetry depending upon the stages. And this figure shows the broad classification of the animal kingdom based on the fundamental feature which we discussed today. So the important characteristic feature of the different phylum are described. So this is all about this session. Okay, thank you very much.